All right, thank you everyone. Thanks for persevering until one of the last talks. We will talk about some data reanalysis, so spiritually similar to what Yves and Yang showed on the ITP, which we also looked at. And specifically, it's about mouse husbandry and the importance of long control lifespans in your studies. And it's work that came to be because Brian from CHL, myself, and Matt Cabell, we sort of had a similar idea at the same time, and then we decided to publish together on this topic. And I'll briefly explain why short lifespans negatively affect interpretation of your results, how long mice should live, like what can we do with this knowledge, and like how can we improve future studies. And I'll start with an intuition going back to human data, actually. Where would you run a lifespan study for a human longevity drug? Probably not in a very poor country where you might have excess mortality due to infections. This could mask your impact on longevity. Would you test your drug perhaps in the US? That might even not be the best place because you have a lot of deaths through obesity or drug-related deaths, so you might be just mitigating premature mortality in a country like the US. Actually, Singapore might be the perfect place to run a longevity study. Why? Because people here live for a very long time, approaching but not quite reaching the biological limits of how long healthy humans can live. So the central message that I want to start with is you want to work in a very long-lived population in both mice and humans if you want to study aging. And in those long-lived populations on the cohort level, you get this compression of morbidity and the survival curves become more rectangular. We see this in mice, we see this in humans, as you see on this slide. So over time, people got healthier and we get these more rectangular survival curves. And now I'll get back to model organisms. So it applies to, different species. Basically, if you look at different strains or cohorts that vary in their lifespan, you plot lifespan on the x-axis versus benefit from longevity treatment, you see that longer-lived species benefit less, or longer-lived strains. And we find this actually in not just, what's happening here? So we find this in mice, in flies, worms, and yeast actually. So if you impose caloric restriction, the longer-lived strains benefit less. And this seems like it would settle it, but actually it's more complicated than that. You have to consider certain statistical biases in this kind of analysis. One is regression to the mean. It's famously important when you run controlled trials. That's why you run a placebo group. But imagine you sample from a population with mice that has a defined lifespan. You have a certain medium-sized cohort, and it's very long-lived. Then the next cohort you will sample will be most likely closer to the mean, and this will affect your results. So we also corrected for this in our published work. So this is from one of those studies where the caloric-restricted mice benefited less if they were from a long-lived strain. And we also showed that at least in this study, this was independent of this regression to mean artifact, which you can test using methods like how you basically ask how would the placebo-treated control group respond in terms of regression to the mean, and then compare it with the actual data. But I don't want to go into too much detail here. Basically, the idea is that short-lived strains will exaggerate your results. So now we need to establish how long should mice live. And under optimal conditions for black 6 and het 3 strains, they live close to 900 days. So we term this rule of thumb the 900-day rule. And if you have an intervention and it leads to lifespan substantially above 900 days, you know it's most likely robust, even if your internal controls aren't good, because this is very, very unusual for these kind of mice to live that long. And once you notice, you can go back to the literature and annotate it with lifespans and try to find promising drugs. And just very briefly before we do this, some people said this rule is too strict, but there are actually many labs in the world which consistently have very, very good lifespans. So I think we're getting there. And yeah, we tried to identify really promising interventions using this 900-day rule. And we can, for example, see the telomerase activation or rapamycin feeding in mice when you start pulling literature data from many cohorts, you see that many of them live longer than 900 days. But this is not true for metformin, which is known not to extend lifespan in mice, so it's sort of a negative control. It might normalize longevity in an unhealthy strain, right? But it doesn't seem to really extend lifespan above 900 days. And you can go to check many other interventions. As mentioned, we have the drug age database, we have gene age for genetic interventions, and you can find candidates that are really promising for just to point out one or two. So for example, L-deprenyl actually works very well in long-lived strains, and it has been picked up by the ITP. It will be tested soon, so we're going to see whether it replicates in het-free mice. Looking at genetics, I'll give you my favorites. For example, FGF21 transgenic mice, they are really unusually long-lived in absolute terms, beating the 900-day rule. So this would be, for example, a promising follow-up. Or NDGA, for example, it works quite well in long-lived black 6 mice and was tested with, as we've seen, some success in the ITP. So, okay. So maybe. And dwarf mice are also a famous example. We know they're unusually long-lived, even if the internal controls in these studies are not very healthy. You see that they're substantially longer-lived than those 900 days that we propose as a standard. And you can start asking the question whether so you can start asking the question whether results in long-lived black 6 mice also predict success in the intervention testing program, which is known to be rigorous. We don't quite have the data to really answer it, but it does seem like there's a trend, I think, as Matt Kabeline pointed out. And what do we do from here? So we've shown that husbandry improved over time. Now it's close, the lifespans are close to 800, 850 days for mice. And we think this is important. This means our studies are more robust than they used to be. And I think we can do even better. And how can we do better? Just very briefly, you need healthy mice. You want them to be specific pathogen-free. So this gives you very, very good lifespans. And going from here, you can also extend this rule, for example, to invertebrates. There should be also standards for how long very healthy invertebrates should live. You can use it to identify compounds and provide additional evidence, retest them. And having access to the ITP data, so for the last 2 slides, you can start doing really cool things with the raw data. Joe Pedro's group recently published how weight affects the results in the interventions testing program. But you can also start asking, could you, for example, replicate the ITP using smaller sample sizes? So on the last slide, I want to ask the question, why isn't there such a thing like high-throughput screening in mice? We know Aura, for example, is doing it in worms, but we have nothing like that in mice. And you can, for example, use the resampling approach and look at the interventions testing program and find that when you resample with between 1 to 10 animals, you already get a pretty good ability to replicate the actual findings of the ITP, which suggests that there's perhaps room in the field to run a large study where you have hundreds of thousands of compounds tested with small groups in mice, and this gives you then preliminary data to follow up on. So that's just as an idea. Maybe someone wants to fund this. We would be happy to run it. And the last slides are just acknowledgements. So thanks to my lab, Brian Kennedy, everyone who supported us, our funders, and for those who have time in the evening, we're hosting a networking dinner and Bella Pizza at 8 p.m. So feel free to come. And are there any questions?
Very nice and important topic. I think I also asked this to Matt, but I was wondering maybe you can look at that if you can look at any association between legislation and lifespans, like in certain countries you have to be stricter with your mice than in other countries and stuff like that. Is that something you could look at?
Yeah, I heard this a lot, and I don't know how to investigate it. I do know that one of our Italian collaborators has fantastic lifespans. I don't know how strict they are. I think you're alluding to euthanasia laws in humane, euthanasia in mice. how strict the laws are there. I know there are European labs that have really high lifespans, but it would be actually good to systematically look at the impact of this.
Hi. So do you have a separate rules for females and male mice? Because when I looked at NIITP data, I noticed that male mice have shorter lifespan, shallower curve, which in our terminology maybe means that they're thicker, and more variable across sites. So maybe there are much more room of improvements for male mice.
Yeah, we didn't look at the shape, but we had a lot of discussions about the male-female difference, and I found that when you have medium-sized data sets with only dozens of studies, you sometimes see differences in black 6 mouse, male and female survival, but once we pooled like 100 plus studies, the differences, at least in median lifespan, vanished, which is consistent with what Steve Alstead showed. That's what I can say.
Hey, that was really thought-provoking in a good way. I was thinking, though, wait, shouldn't we also want to run clinical trials in all those countries so that we can benefit lots of different people? So even the short-lived ones, you know, things that we can find out in short-lived mice or whatever could have a benefit there. So I'm wondering, like, maybe using like diversity outbred, like increasing the diversity, but also increasing the number of concentrations of drugs so that you can really increase the effect, because something might fail if you've only got one concentration, but was it the right? Did it get into the tissue? Did it do what it needed to do? There's probably lots of thoughts in that question then, I apologize.
Yeah, absolutely. I mean, they should also work in other populations, and it's worth studying. You just have to, you know, the funds are limited, and you have to decide how and where you do your study.
No, I appreciate that.
Yeah.